You're listening to the Good Question Podcast with Richard Jacobs. Our goal is to make each of our guests exclaim, that's a good question. I don't know the answer. Because when that happens, it means you, the listener, may be inspired to learn more beyond the interview and to ask great questions yourself that lead to new insights. In this podcast, we cover historical and current anthropology, comparative religion, and history. Welcome, and let's get started.
Hello, this is Richard Jacobs with the Good Question Podcast. My guest is Shemesh, PhD. He's a principal investigator. He has his own lab called the Shemesh Lab at University of Pittsburgh. We're going to talk about something interesting, infectious neuroscience. So this is the ever-growing likelihood that certain diseases we thought that may not come from an infectious source actually may. Alzheimer's, other neurological disorders. So I think this will be a very interesting call. And welcome, War. Thank you.
Thank you. Happy to be here. Hi, Richard.
Tell me first a bit about your background. How did you get into the area you're currently studying?
I grew up in Jerusalem, Israel. When I was about 22 years old, my mom calls me and she says that grandpa has Alzheimer's. He was diagnosed with Alzheimer's. And one of the first things I did, I went and spoke with his PCPs. Spoke with his medical doctors, the people who were supposed to take care of him. That person told me it's a disease where you have inflammation in the brain. It's a disease where there's no any known treatment that can reverse Alzheimer's, and there's not going to be a cure in 100 years with these words. And that spiked my interest and pissed me off too at the same time. So that's how I got into.
You took it as a personal challenge to do something. It sounds like
it is a personal challenge. The problem is that he was diagnosed, I think, around 2000, and my grandma was diagnosed with the same disease She was diagnosed in 2012, I think.
Okay.
So, and the treatment, the prognosis, the treatment, everything was the same. So, literally within the span of the decade, with all the billions of dollars that have been poured on studying Alzheimer's disease and the trillions that have been poured on treating Alzheimer's disease, nothing really improved substantially. So that is the grim world we live in today.
Okay. When and how did you make a connection that there could be an infectious source of Alzheimer's instead of an endogenous source, you know?
Yeah. So, by the way, I'm not only in the University of Pittsburgh, I'm also in the Hebrew University of Jerusalem in Israel. But related to this connection, very early on, I saw what I call red flags. And I studied Alzheimer's disease for my PhD. As exciting as it was, I always felt something was missing. For example, it's a disease where you have inflammation in the brain, as this very wise doctor told me at the time. What's the major cause of inflammation? There are many causes of inflammation, as we know. There's inflammation that comes from the gut, from what we eat, but the major cause of inflammation, first and foremost, are bacteria and viruses and yeast and whatnot. The second thing that spiked my interest is the fact that I started to see, I read a lot of papers, and in some of them I saw interesting things like if within a couple, married couple, if one of the persons has Alzheimer's disease, the spouse has a between 4 and 6-fold increased probability of having Alzheimer's disease. That was actually a very big red flag for me.
What was the flag? I'm sorry, I'm not clear on
the fact that there were studies conducted showing that if within a couple, if one person has Alzheimer's disease, the spouse has a chance that's 4-fold to 6-fold higher in having the disease in their lifetime.
Why?
And that's the answer to the why, because it's infectious. As you know, we humans tend to exchange liquids when they're married, and they tend to live in the same area, eat the same food, and prosper in the same or not in the same environment. So I really didn't find it. Was so clear to me that something is amiss there. Also, what I noticed is I read papers that show that Alzheimer's most of the time doesn't come alone. I mean, it's supposedly the harbinger of death, but most of Alzheimer's cases come with comorbidities. Alzheimer's and another disease of the nervous system, Alzheimer's and two other diseases of the nervous system. Alzheimer, for example, Alzheimer and ALS, Alzheimer's and Parkinson's disease, and Alzheimer's and CTE. And that can, around only 30% are only Alzheimer's. And that can point out that there are discrete causes to different diseases. And when they come together, they manifest as multiple diseases, multiple separate diseases with separate symptoms, and that are stereotypically known. And these discrete causes could be pathogens. And I also was exposed at the time. So whenever I spoke about it, people laughed at me. I was a PhD student. No one took me too seriously. But then I found the papers by Professor Ruth Itzhaki from the UK, and she was working on it from the 80s to 90s. And what she found was a connection between herpes simplex virus 1, the cold sore virus, and Alzheimer. And in a slew of papers, she basically created the web of connections between very early papers that were kind of overlooked. So this is how I came to learn about this unique connection between disease of the nervous system and infectious agents. And this is what we actually do in my lab today.
But how did you even get this idea, and how would you identify which infectious agents, a virus, a bacteria, how did this even come into your mind?
This is a great question because it was all out there, meaning that you can figure this out by looking in the literature, or you can figure it out in a non-biased manner. And what do I mean by looking at the literature? From the 70s, there have been instances and case reports, basically people coming to the ER and later to the OR with Alzheimer's disease. And they were tested, meaning that the blood was drawn or saliva was taken. And when they were tested and analyzed, they had a much higher incidence of encephalitis or viral encephalitis and or just viral infection with herpes simplex virus 1. Similar to these cases or case studies have happened with microbes or bacteria, such as a chlamydia. The chlamydia bacterium is a mostly intracellular bacterium. There's chlamydia pneumonia that causes pneumonia, of course. There's chlamydia trachomatis, and that's an STD. They were found in samples from patients by Professor Brian Bailey from PCOM in Pennsylvania. So basically what my group did, we looked over the literature and we said, okay, we need to label these things that appear in the literature, HSV and Alzheimer's, and herpes simplex virus 1 and Alzheimer's, and herpes simplex virus 2 and Alzheimer's, and the chlamydia pneumonia and salmonella and Alzheimer's. And then we needed a way to label them, all of them within the brain tissue. The problem in brain tissues, human brain tissues from the elderly, is that basically you go to a brain bank and you get a brain bank is basically where the family of a patient donates their brain to a facility, and you look at postmortem samples. The problem there is that when we age, we have aggregation and inclusions of oxidized proteins in our cells, and oxidized lipids and oxidized DNA and RNA, and they are superfluorescent, meaning that if you look at on them under microscope, everything looks positive. Everything, it shines in all colors. So you can't really label anything. It's basically noise. So we figured out when I was in MIT for my postdoc in the lab of Professor Ed Boyden, they invented a method called expansion microscopy. What is expansion microscopy? It's a super resolution technique where you can look at things that are really, really small at 40 nanometer resolution.
Wow. How does it work? How are you able to see things at that? I've heard of like cryo-EM, but how does it work?
Yes, so the cool thing about expansion microscopy is that you don't try to play with the physics of the super resolution. It's a tissue expansion method. You take the tissue you infuse a diaper polymer into the tissue. You get a molecule that binds between the diaper polymer and all your proteins, and it's like an anchoring molecule, and then you cook it for an hour at 120 degrees with very, very strong reducing agents and denaturing agents. And basically what you get at the end is a gel. It's a hydrogel. That it's like the same hydrogel that you have in a diaper. When you add water to a diaper, it swells up. When you add water to the gel, it swells up by a factor of 5 approximately. So the tissue is expanded now after I added water by a factor of 5. Now I come with the same microscope that I imaged or used before, and I can see things at a resolution that's 5 times better. Not only that these oxidized proteins and oxidized lipids and everything that stood in our way is gone. So now whenever you see a signal, it's for real. This is a positive signal and not an artifact. And we were able this way to look at all of those pathogens in the brain at super resolution, at 40 nanometer resolution. And what we saw was pretty striking. You got to see immediately HSV1 and HSV2 proteins and chlamydia and candida and salmonella. So it was a big...
in the brain.
In the brain. Yeah.
Are you saying that certain infections that can go into the brain then can cause a cascade over X number of years to cause Alzheimer's or some of these other brain conditions, Parkinson's?
Yeah, absolutely. The answer is that it's complicated. It depends on the pathogen, meaning that some pathogens go in pretty early and lay dormant. They become latent. For example, the herpes viruses are. And some pathogens, when they enter the brain, they enter at a completely different stage of the disease, meaning that the herpes could have been there for 40 years and the salmonella just entered later on in life and they wreaked havoc. So different pathogens come in at different times of the individual's life.
Are you able to tell which pathogens will lead to which conditions? Is there any direct correlation yet?
Yes. So what we do and others. So I can't discuss exactly what pathogens because we're working with a lot of other investigators in collaboration. So I still don't have their permission to talk about everything, but I can tell you that we definitely see a very, very strong correlation between a disease and its microbiome or its brain microbiome or pathobiome, meaning that Alzheimer's has a microbiome on its own, and Parkinson's has a microbiome on its own, and CTE, chronic traumatic.
Wait. So when you say microbiome, are you seeing? Well, first of all, have you looked at what you consider to be healthy brains, and are there bacteria and other microbial constituents even in a healthy brain?
Yeah, it's a wonderful question. So absolutely, in the healthy brain, in most cases, we do not see the pathogens at all. In some cases, such as in herpes simplex virus 1, we definitely see, and that for me, it was a shocker. Every brain that I looked into, whether they were control brain of people of the same age and the same biological sexes and post-mortem interval, every brain of the control and of the Alzheimer's had herpes simplex virus 1. The difference was how well spread it was, meaning that with Alzheimer's disease, the area that the HSP1 proteins were occupying was much, much larger compared to Alzheimer's.
Well, all right. So, question here: you know how there's leaky gut syndrome, which allows stuff to get into the outside of the digestive cavity. Is there? Do you think there's an analog here, leaky blood-brain barrier syndrome, that allows for this?
Yeah. So if we're talking about how they get into the brain, the gut or the leaky gut is definitely one way that the gut microbiome can erroneously get into the brain. So there are a lot of things that cause our guts to get leaky and have holes in them, such as, for example, lectin proteins that exist in nightshade vegetables, for example, and many, many other things that cause inflammation and leaky gut. And then things like candida can end up in our brains. We see a lot of candida albicans, that yeast fungus inside the Alzheimer's brain. So this is one way to get there. If we're talking about ways to get there, and there are labs that work on it, there are a lot of different ways, but at the end product or the end point is a brain that acts like a sponge, meaning that it's not so easy to get in, but you can get in. But once you're there, the blood-brain barrier locks you inside. So it's like a sponge.
Does the one-way barrier appear to be characteristics of a one-way membrane, or is there, what particles or compounds tend to come out through the blood-brain barrier? It's not all one way.
Yeah. So there are a lot of ways where the blood-brain barrier, things flow in and out, and they're different. When blood-brain barrier fails, things can get in, and we think that one instance of one disease that's a classic example of that is CTE, chronic traumatic encephalopathy. The disease is not well known in NFL players, for example, Arnold Hernandez. We all saw it in the public literature, New York Times papers and whatnot.
Well, maybe a concussion temporarily compromises the blood-brain barrier, allowing stuff to get in there.
Exactly. So what did we do? We got brains from the Boston Brain Bank. That's a brain bank dedicated to CTE, NFL players, and contact sports athletes like boxers and UFC fighters. And we compared folks who did post-mortem brains of people who have received a lot of head blows and didn't have CTE, and received head blows and didn't get CTE. And the difference is in the pathogens. There's way more pathogens, and the spread of the viruses is much larger with people who have CTE or diagnosed with CTE. So what we're testing now is whether the blood-brain barrier, falls apart momentarily, and then again, and then again, and then again, multiple times, and circulating viruses, for example, or bacteria get into the brain and stay there. The other thing is that viruses can get activated by getting these head blows. They lay dormant, as I said before. They can wake up. They can become active again. The infection becomes active. And then the more you get, blows you get, the more cumulative damage of activated virus you get.
All right. So if I'm a football player, for instance, if I have a really good diet, I don't have leaky gut, I get a concussion, I'm probably less likely to have CTE later on versus a football player. Maybe they're a front linesman. They're real big. They don't have the greatest diet. They got a lot of extra fat and all the other stuff going on, and maybe they have leaky gut. So they get a concussion. It may be more likely that's like a nucleation or a seed that would get into their brain to cause trouble later on.
Absolutely. And the opposite is true too, because I've been trying to get in touch with the NFL lately because we can solve a lot of problems to the sports practitioners and the teams. If indeed it's caused by pathogen, all you need to do is have some prophylactic measure or preemptive treatment with a pill.
What about an immediate post-concussion something that would shut the blood-brain barrier instead of letting it open up? Maybe a cold applied to the site for a period of time, or some other protocol that may generally prevent the introduction of this stuff.
Yeah, the problem is that with compromise of the BBB, it can be very, very deep inside the brain, so you don't really know where to go. You can have several types of medicine. One could be immune boosters, something that will work on the immune system in the brain that will boost it against these pathogens. And the other would be the antiviral, antibacterial, antifungal drugs.
Do you think, so then let's say Candida, whatever that gets into the brain, now it's not in its native environment. It has its own nutritional needs. It produces waste, these different bacteria. Is that possibly the source of certain plaques in the brain? Maybe that's the waste from certain bacteria that are dying or metabolizing in the brain. Does this compromise glucose uptake and utilization of the brain because now you've got bacteria there that are feasting on the glucose versus the brain?
Absolutely. There's a lot of studies now that claim that Alzheimer's is a type of a metabolic disease, and that we basically lose the ATP. We lose this universal coin of energy. But if you have these pathogens in your cells, a lot of them are intracellulars, a lot of them are in between cells. They hijack. They come and they basically burglarize everything. They come, they steal away the energy. So your nerve cells are left behind. They can't supply the huge amount of energy that they need to fire. That's what brain cells need to do. The other thing related to what you're saying is two more things. One is that, for example, a candida can, we know that a fungus can lead to hallucinations, and one of them is candida. They secrete materials that can lead to hallucinations. Last thing is that what we're seeing in our studies is that the pathologies of Alzheimer's and the pathologies of Parkinson's and whatnot, they colocalize very strongly with the pathogen. A-beta is actually beta amyloid protein. The extracellular pathology of Alzheimer's is an antimicrobial strategy, and tau is an antimicrobial strategy, and alpha-synuclein is an antimicrobial strategy, and CDP43 is an antimicrobial. And there lies the big tragedy in this field, which is everyone, but a lot of the scientists are trying to kill them off.
What about
anti-tau drugs, and so on and so forth. We need a new class of drug.
Well, all right. So what I would expect is, are you looking at candida in, the gut environment versus the brain environment? Maybe you could use two different organoids and Absolutely. Look at the difference, the epigenetic marks that would appear with these bacteria in an unfamiliar environment, the brain. Therefore, their metabolism will be different. The waste they produce is different, et cetera. Has that characterization been done? And if so, has that revealed anything interesting?
So the answer is partially, because it's complicated. It's complicated from the technical issue that we have is it's sometimes you don't get the brain and the gut from the same person. Because when the brain bank is built up, most of what they have originally is the brains. And then they may have the blood samples from the same person. And then they may have CSF, cerebrospinal fluid, and then they may have saliva and whatnot, and maybe they'll get something from the gut. But if you look at how two things behave this way or that way, you can learn a lot. You can learn tons about what happens with the pathogen when it gets to a terra incognita. And related to that, what we see in the by the way, we sequence. We do metagenomics. We look at the genes. And we do transcriptomic sequencing. We look at the RNA in and out of the brain, meaning in the blood versus the brain. And we can see that the blood and the CSF is only a proxy. It's just mildly representative of what happens in the brain. And the brain has its own story, and the blood and the gut and everything is a completely different environment. That's why, and for many years, people have been avoiding talking about the brain microbiome, but it's a real thing. It's a vast thing, by the way. We found hundreds to thousands of microbes within a brain, whether it was sick or healthy.
All right, so that's what I asked you before. So in healthy brains, you do find there is a pre-existing brain microbiome.
Absolutely, but the microbiomes are different.
Okay.
Meaning, got the same baseline. I'm sorry if I wasn't clear about it. I'm so excited, but there's the same baseline. And then in Alzheimer's, you have certain microbes that are elevated. Then in Parkinson's, different microbes that are elevated. And it's not necessarily one microbe. Meaning that one person get Alzheimer's, it can be caused by 4 pathogens and another by 5 or 6. And they can be mostly different. They can be mostly different. The other thing is that the less patients a disease has, and the more stereotypical it has, the less types and the smaller number of species we find.
Meaning in healthy brains or sick brains?
In sick brains. meaning that
you have more diversity in sick.
Alzheimer's. In Alzheimer's, you have more diversity because
So
Alzheimer's
usually the gut is the opposite.
yeah, correct. The brain is not operating by Occam's razor; it's operating by Occam's anvil. Everything is very weird there.
So in the gut, I've heard from many microbiome scientists, more diversity is better. That seems to be the supposedly common knowledge. The brain, though, it seems like the opposite from what you're saying. Less diversity is correlated with healthier brains, or no?
Not necessarily. The diversity is what we were saying is pretty similar. It's specific species of bacteria, of viruses, of different types of yeast that are elevated in diseased brains. But in Alzheimer's, where you have 35 to 40 million patients, you have a pretty large, dozens of species, disease microbiome that characterize the diseased brain. In ALS or CTE, you have one or two that we can find. So that are characteristic of the disease. And in Parkinson's disease, where you have 6 million patients, it's not 1 in 50,000 like in ALS, and it's not 40 million like Alzheimer's. It's somewhere in between.
So, all right. Okay. All right. I got you. Have you looked at, so any of these bacterial species similar to what's in the gut? Are you able to name any of the common ones that show up for different conditions?
Yeah. What we find is that some of them exist in the gut, and some of them, the things that we find through sequencing. Through sequencing, you see pretty insane things, because we are trying basically to mine the genes and the RNA that could have accumulated for an entire lifetime. So basically what we see is that a lot of bacteria and parasites and parasitic bacteria that infect animals. So if you're an animal lover, wash your hands. And we find bacteria that live in puddles, and we find bacteria that live we find microbes that live in sewers. If I have viruses...
Oh, I'll give you a closer example to home. I had interviewed a few years ago Florencia McAllister. She was studying the microbiome of pancreases, and she saw that where there was cancerous nodules, it had its own particular microbiome versus a healthier part of the pancreas. So it looks like, based on your research, that there may be microbes in every part of the body, and nothing is truly sterile.
Nothing is truly sterile, and we need to keep the microbiome healthy. I do not know, based on our studies at this point in the scientific history, whether there's such thing as a healthy microbiome in Alzheimer's, a microbiome in the brain, and a microbiome that you have to maintain and to feed, like with prebiotics and probiotics in our gut. Still, we don't know that. We know that there is a microbiome that's bad for us in Alzheimer's, or there's a very strong association between that and Alzheimer's. And this is, by the way, the reason that all of those lifestyle gurus and all of those lifestyle studies came about. If you run and swim, the chance of Alzheimer's drops by X%. Why? Because your immune system it all fits. Because the immune system is fortified when you do exercise, and then you're less prone to infections. And the other way is, and the risk factors, it's the same thing with the risk factor. You know that for Alzheimer's, head blows are risk factors. That's what happened with my grandpa, by the way. He was involved as a pedestrian and hit and run. And I think it was very lucid. 3 months after his accident, he started having issues.
No, I've had 2 concussions. My brain was foggy for a couple of weeks after. But when I did a significant amount of exercise, I literally woke up and I felt, and my mind felt clear. So maybe there's some correlation there. But that I can tell you that was my experience. I felt and I couldn't think clearly.
Yeah,
and then, about an hour of continuous exercise cleared it.
Absolutely. And we had it with COVID too, by the way. All this brain fog and long COVID, I think we'll see a spike in dementia.
What about people that get mono or encephalitis? that I guess could set them up for problems from then on.
Absolutely. Mono is Epstein-Barr virus, EBV, and mono has been linked to another disease very strongly, which is multiple sclerosis, MS. And there were papers in Nature and Science about that. It's I wouldn't say it's all but accepted, but it's more it's getting into consensus, becoming consensus, slowly but surely.
Have you is there any I know there's so many studies to do, but is there a possibility of doing a study where people have early, they're early stages of Parkinson's or Alzheimer's, and they go on a strict diet protocol and exercise regimen? Is there a correlation in the reduction? I guess I don't know how you'd sample their brains either.
But yeah, there are a lot of studies these kind of studies. There are a lot of studies of this sort, and you always see a correlation between better lifestyle and lower incidence of Alzheimer's. It's so murky. There are studies, for example, in that there's an ongoing study now with mild cognitive impairment patients, basically very early stage Alzheimer's, where they get cyclovir, the HSV1 medication. And the study is still ongoing to see whether it could ameliorate the symptoms.
When you're looking at the brains from the brain bank, is that a microbiome, or really maybe it's a necrobiome because the person is dead, the brain has been dead for a while? Could there be significant change from someone being alive versus being dead?
Absolutely. So this is a very, very interesting topic and a very, very interesting criticism. And I call this the bone saw fallacy, meaning that a lot of people ask us, hey, in pathology, basically they come in and they saw the brain with a bone saw. How do you know that from that filthy bone saw, microbes didn't fall in the brain, then it stayed there for another hour or two, and then it's full of microbes? And there are a lot of answers to that. So I'll go through them one by one. First, this particular infestation of the brain post-mortem happens for some incredible reason only in Alzheimer's brains. You don't see them in non-Alzheimer's brains. We made sure that the treatment of the Alzheimer's brain, to the best of our ability, of course, you can never be too sure, that the treatment of the Alzheimer's brains and the non-Alzheimer's brain in terms of post-mortem interval was similar and as short as possible. There was not enough time to have a brain infestation at that scale. So that's the first thing. The second thing is of course that there's a big difference despite the same treatment between Alzheimer's and between disease and non-disease. Let's call it like that. The second thing is that we see exactly where these pathogens are, the pathologies of Alzheimer's disease. We see a co-localization between wherever you have A beta, you have a microbe. Physically, it's on it. So it's just a wonderful coincidence if it's just associative and not causative. We can talk about causation very soon. And the last thing is that when we take the brain, when we cut the brain, the third thing, when you cut the brain, you make sure that you cut it centimeters away from where the bone saw hit, meaning that it's impossible or highly, highly improbable that within the 6 hours or the 10 hours or the 12 hours that the brain was in the post-mortem interval, that the pathogens have filled it to the brim.
Interesting. Well, are you doing any work with, in order to I know you can't sample people that are alive for the most part. What if you, again, created a brain organoid and, we're able to test it this way to see what kind of uptake there would be of candida and herpes, et cetera, and what kind of localized microbiome would evolve?
Yeah. We actually do that. We do that with 2D cultures ourselves, and we also collaborate and do it with 3D organoids, brain organoids, and we collaborate with Professor Leonardo D'Aiuto from the University of Pittsburgh, from here or there in Pittsburgh, and basically it's a model, and the model is that you scrape off cells from our epithelium, and then you de-differentiate them then into stem cells, then you make them into neurons and you grow them, and then eventually you have this small organoid that's less than a millimeter or a millimeter big, and it's supposed to recapitulate some of the. As you see, You see, you hear the hiccups. I'm skeptical, but it does recapitulate some of the biological process that happen in the brain.
Well, some, right. Another approach that I thought of is looking at CSF, the cerebral spinal fluid, that you could, theoretically get from people if there was another condition that required, let's say, a spinal tap or something.
Exactly.
That may, the bacteria and various metabolites may show up there, and instead of now looking at what might be a massively altered microbiome become necrobiome, maybe now you get a truer approximation of what's going on.
Absolutely. The issue with spinal taps and the issue with CSF in the States is I've been trying to get them. And it's always the same story. You reach out to a brain bank and say, yes, we have the brains for you. And then we say, great, because you can get them paraffin embedded and formalin fixed. And then I ask, hey, do you have them frozen? Because we also we sequence the brains. We look at DNA in frozen brains. Say, yes, we got it. And then I say, do you have blood? And then some of them say yes and no. And I said, do we have CSF? And they usually don't have CSF. In Europe, I think lumbar puncture punctures are more common. Maybe I'm wrong, but there are more samples from Europe. So we're trying to get it from European brain banks versus American brain bank.
Well, I mean, if they have enough, it's not a nice thing, but if a brain bank has enough volume coming in on a regular basis, I guess what you'd have to do is try to set it up where, next month's brains, we want 10 of them, and make sure you keep the CSF and other stuff. Don't process them and give us fresh brains unprocessed.
Yeah. No, I think we need a standard protocol, 2 standard protocols. And one is for getting samples or harvesting samples for a brain bank, getting the fixed samples and the frozen samples and the blood and the saliva and the CSF and whatnot. And the other is figuring out how to nail down which pathogens are in there and which pathogens are correlated or causally linked to the pathology, which is part of what we do. I'm part of a group, a consortium of sorts called ALZPI. It's led by Nikki Shutek and Brian Balin. And we're trying to figure out this protocol, this consensus protocol, as we call it, to understand what pathogens are in the brain, in the Alzheimer's brain.
One more thing, I don't know too much about it, but supposedly when people sleep, the brain tissue will shrink or move to allow channels to open or maybe expand to allow drainage. So when you talk to the brain bank, what might be interesting is if you can get brains of people that died in their sleep versus people that died while awake.
That's interesting. I like it. Richard, I like it.
Yeah, there might be a very different, things going on at that point.
Yeah, absolutely. There's also a very valid criticism that we have to take into account when you look at Alzheimer's brains. In many cases, where from the diagnosis into bed and into mortality, there's an average of 7 years. And in these 7 years, there's at least 2-3 years where a patient is sitting in a room, bedridden, and looking at the walls. And that affects greatly the brain, the number of synapses, basically preventing these people from very precious stimuli. And that could affect the brains too. So this is something we also have to take into account when you compare the brains.
What about the condition of the blood-brain barrier, the integrity versus age? A lot of these conditions, again, take years to develop, and therefore they tend to skew in older people, unless you get early onset. But, has anyone done a study on looking, over different ages, the state of the blood-brain barrier? That may inform what you're doing.
Yeah, absolutely. There are a lot of studies on age versus BBB integrity, and it falls apart. But basically, we have at certain ages we have holes that are quickly closed and sealed, and in later ages you have permanent leaks. For example, if we look at brains of elderly people, or we look at certain elderly people, some are intact, and people with Alzheimer's, for example, you see we have markers. We have specific markers for a leaky BBB, or basically bleeding into the brain. And the brains of Alzheimer's patients are bleeding in from the outside in. It's terrible, but it's very telling. Also, look at the trajectory of Alzheimer's. Alzheimer's is never linear. It's not a monotonic decrease in abilities. It has quantum leaps, so to speak. Step function changes, meaning that a person has a slow decline in their abilities, in their cognitive abilities, and then, boom, they can't walk. A week before they could not run, but could walk quickly, and then they're bedridden in a week. Or they become really disoriented in a matter of 2 months. And the famous stages of Alzheimer's, and not talking about the Bragg staging, but the behavioral manifestations of Alzheimer's disease. So it's not a linear thing. There are changes that are robust, and these changes could be explained through pathogens, meaning that one pathogen or one failure of we can look at it either from the pathogen side or the immune system, but the immune system can't hold it at bay for enough time. Then it falls apart, the BBB opens, everyone joined the microbiome palooza, or at least a certain cabal of pathogens get into the brain and wreak havoc. They lead to the mortality of cells and lead to the famous and well-established pathology of Alzheimer's. A-beta, as I said, is a antimicrobial strategy, and tau could be an antimicrobial strategy. But if you use them too much, they become toxic. We know they become toxic. There are 50,000 papers about A-beta and 25,000 papers about tau. I know, I wrote a very, very small portion of them. But after a certain amount of insult, these innate immunity factors become detrimental. They become part of the problem. So basically, if we're trying to design an Alzheimer's drug, we should design things that would do what A-beta and would do what tau does, only without the side effects.
Interesting. And I wonder what the interaction between the beta and tau plaques are with the existing, microbes that are in there.
Yeah.
Are you able to ascertain that? Do they feed off the plaques? Maybe they create a biofilm on the plaques that's different? I mean, in their presence, I wonder what happens.
Absolutely. We can tell that the oligomers, more than the plaques, are more dynamic, and they are better linked to the pathogens in Alzheimer's disease. But yeah, we have two papers about this coming up.
Well, have you been able to do shotgun metagenomics on the brain microbiomes and to see what what products they may make or what wastes they may generate, consuming the glucose in the brain or whatever they consume?
Yeah. We did the only metagenomic studies that we do. Basically, what we do is we do metagenomics, we sequence the brain, and we do transcriptomics. We get the RNA, and then we analyze, and we say, okay, let's throw away everything that's human, and then we're left with the good stuff. Thousands and thousands of viruses and bacteria. It's just amazing to see how much there is. That said, it doesn't mean that at the time of death they were active, which is the reason that we do expansion microscopy. In expansion microscopy, you can see them, and you can tell based on the protein expression, the proteins that they manufacture, that they're actually alive at the time of death.
Okay. Very interesting. I definitely would love to follow up and see how this progresses. This is very, very interesting. Where can listeners go to find out more about what you're doing and follow the Shemesh Lab?
We have a lab website, shemeshlab.com. That's it. And there you can see what we're doing. Publications are pending. So yeah, I would love to talk more.
Excellent. Very good. Thank you for coming, Or.
Awesome.
Thank you for listening to the Good Question Podcast. Please email support@thegoodquestionpodcast.com if you have any referrals to great guests for us to interview. Visit thegoodquestionpodcast.com to hear more interviews. And please help us spread the word by rating and reviewing us on Apple Podcasts. iTunes, Spotify, YouTube, or wherever you listen to this podcast.